I thank Ramanujan College and Miranda House for giving me such an wonderful opportunity to interact with such a wonderful audience. In this faculty development program on life science, I am going to deliver a talk on development of topical formulation for radiation dermatitis. Myself, Dr. Himanshu Ojha, scientist E, working in Mars, a lab of life science cluster of DRDO under Ministry of Defense. So, before proceeding toward our presentation, I would like to take a short journey about the history of nuclear accident. In 1945, on August 6th and 9th, the Allied forces bombed Hiroshima and Nagasaki with nuclear warheads, which led to the killing of around 4 lakh people. And the world first time witnessed the harmful effect of ionizing radiation. Then in Chernobyl, Ukraine nuclear reactor 1986, a team of Russian scientists were conducting a test, but because of the faulty reactor design and non-adherence to the protocols, it led to a nuclear disaster, which led to the mass-scale displacement of the population of Pripyat town. Still, this area is uninhabitable. There another infamous incident in Geonia, Brazil, where a often so radiotherapy source uh, found in the abandoned hospital site, which lead to the death of around 4 people and lead to the contamination of 249 per persons. In 2010, a place called Mayapuri in West Delhi, where a radioactive source was being procured from Department of Chemistry, University of Delhi. Then the scrap dealers dismantle this radioactive source there and then, which lead to the exposure of radioactivity, and one person died because of this exposure. In all these incidents, the radiation dermatitis was found to be a common symptom, and it is the first symptom to appear on the skin. Why skin? Because skin is the largest tissue of the human body and the most sensitive one. So this is a skin first act of barrier of defense get affected by the radiation exposure. Now let us move toward the other basics. So how the radiation enter or expose our body. So we can see that in a ingestion, the mode where a person having contaminated with the radioactivity, eat it with its infected hand or drink something which lead to the internalization of radioactivity inside the victim. Other is inhalation. Through the nose, a radioactive dust can be taken into the body, which may lead to the further complication. Wounds, where a person is got a injured tissue, where the it serve a purpose to enter a radioactivity inside the body. Other is injection mode, which is very not very common. Now, if suppose a person got contaminated with the radioactive dust, he may spread secondary contamination through cloth and through skin. This process is called external contamination, where our group is focusing and developing formulation to decontaminate it. Simultaneously, we are also working on if radioactivity lead to the radiation dermatitis, how to mitigate or how to prophylactically use some formulation to prevent the radiation dermatitis. Now, let's move to the molecular level. What happens when radiation interact with the biological system? We know that DNA is a critical organelle of human body. It is a storehouse of hereditary information. It is a you know overall um, thing that dictate the human machinery. We know that it's a double strand helix compound which interact with the radiation directly or indirectly. The damage vis-a-vis to direct and indirect damage depend upon the linear energy transfer. That how much a radiation can deposit energy in per unit area. That dictates that how this damage will cause, which mechanism it will follow. In case of gamma, which is a low linear energy transfer ionizing radiation, the damage is mostly through reactive oxygen species, which is generated due to the radiolysis of the water. This ROS interact with the double helix of the DNA and create radical centers, and further it lead to the disruption in the structure. Because of the structural disruption, the very physiological function of DNA got compromised. But there is a machinery in place to repair this DNA damage. But when the burden of ROS becomes so high that it lead to a condition of oxidative stress, then in that case, it underperform the efficiency of DNA repair machinery. In that case, the machine got compromised, and the optimal efficiency of cell goes down. And ultimately, if there is no medical intervention provided to the victim, then the victim may die. Similarly, with the proteins, we also know that proteins play a critical role in the overall human physiology, but due to the interaction with radiation, through the interaction of reactive oxygen species, it lead to fragmentation, aggregation, or lead to the formation of protein carbonation and peroxides. So, because of this, the structural functionality got compromised, and we know that the overall functionality of such structures is depend upon their structure. If their three-dimensional spatial structure got disrupted, then their functions got compromised. Now, let's move to our topic, radiation dermatitis. Now, what is this radiation dermatitis is all about? Radiation dermatitis is a side effect of external beam ionizing radiation. It is also called radiation dermatitis, X-ray dermatitis, is radiation skin damage, or a radiation burn. So it's been known by different names. Most commonly, radiation-induced dermatitis is caused by radiotherapy, which is a planned radiation exposure given to cancer patient. And under this, the doctors trying to treat a patient for its underlying malignancies. It may also rarely result from exposure to radiation during interventional procedures such as coronography, angiography, embolization procedures, inhaling catheter placement. So in all those things, the patient have a possibility of being exposed to ionizing radiation. A radiation of the skin lead to a complex pattern of direct tissue injury, which involving damage to cell of epidermis, endothelial cell within the wall of blood vessels, and inflammatory cell recruitment. There are certain clinical features by which radiation dermatitis can be characterized. The radiation dermatitis generally manifests within few days to weeks after the start of radiotherapy. Its onset depends on the radiation dose, intensity, and the normal tissue sensitivity of the individual. It also confined to areas of skin that have been irradiated, and skin changes are sharply demarcated. The radiation-induced dermatitis can be classified as acute or chronic. Now, what is the type of these radiation dermatitis mean of? The acute dermatitis is basically generally manifest within few days to few weeks after the exposure of ionizing radiation. It present as red patches with desquamation or blistering. So desquamation is just meaning of peeling of the skin. In the chronic radiation dermatitis, which uh, occurs with the chronic exposure to low doses of ionizing radiation over a prolonged period of time, producing varying degree of damage to the skin and its underlying parts. There is another third type which is called delayed types. In the delayed type, the radiation acana occurs at site of previous ionizing radiation exposure as the acute phase of radiation dermatitis begin to resolve. In the acute radiation dermatitis, the time frame is 90 days of the exposure to radiation, and the patient may have skin changes ranging from faint edema. Edema means reddening, and the desquamation means peeling of the skin. Then it leads to the skin necrosis or the death of the skin cell, and finally the ulceration, depending upon the severity of the reaction. According to National Cancer Institute of USA, has developed a four criteria for the classification of acute radiation dermatitis. In grade 1, the faint edema or desquamation take place. In grade 2, the severity increases to moderate to brisk edema or patchy with moist desquamation confined to skin fold and creases with moderate swelling. In the grade 3, acute radiation dermatitis, confluent moist desquamation, which is greater than in size of 1.5 centimeter diameter and which is not confined to the skin fold, pitting edema, severe swelling. In grade 4, the skin necrosis or ulceration of full thickness dermis take place in the middle layer of the skin. So, in the chronic radiation dermatitis, the period of onset is may occur from 15 days to 10 years or more after the beginning of the radiation therapy. It is an extension of acute process and involves further inflammatory change in the skin. The chronic radiation-induced changes in the skin are characterized by disappearance of follicular structures, pores, an increase in the collagen and the damage to the elastic fibers in the dermis, a fragile surface skin, and telangitis, which is a prominent blood vessels. In the telangitis, the it lead to a construction of the blood vessels, which lead to a patchy, spidery appearance of the blood vessels, and because of this, it lead to a further complication in radiation dermatitis. Secondary skin cancer may also resulted from the repeated exposure to radiation therapy. Mostly common is the basal cell carcinoma. We can see from this cartoon there a normal skin which is been into divided into epithelial, dermis, and the subcutaneous part. Then an insult happen with the exposure of radiation, and it lead to the enhancement of reactive oxygen species TGF beta and TNF alpha. Then, because of the interaction of cytokines, the injury part is getting the required compounds which required to be heal up the tissue part, but there is aberrant healing fibrosis. If the machinery is overwhelmed with the reactive oxygen species, then the it lead to a complex process and the healing process got stuck up. Therefore, it's a challenge to, for the clinicians and scientists to develop topical formulation so that we can heal up the things in a well-defined time frame so that a patient may not lose its quality of life. So this is the some pictures which I have collected from the internet from different sources that how this radiation dermatitis appear. We can see that in the neck region of this patient, male patient, in the neck region, the appearance of the erythema. Then on the back side of this patient, we can see the grade of this acute dermatitis. Then in the long run, that how this dermatitis look like in this patient. So these all pictures manifest the different appearance of radiation dermatitis. Now the lay of my scientific presentation is like this. So I'm going to introduce you that we have a compound called ethyl pyruvate, which is an anti-inflammatory compound, and how this anti-inflammatory compound have an anti-radical potential. Then how it assess its potential, that it protect DNA and the protein at molecular level against the radiation-induced oxidative damage. Then we cannot apply this molecule as such, because always when it is for the clinical purposes, we need to formulate this compound in order to enhance the solubility and permeability in the inside the skin. So we have to go for a formulation which is to be stable, which is to be a the well characterized one. And here we have developed a liposomal hydrogel formulation. And why hydrogel? We will explain in our later slide why we have gone for the hydrogel. Then, before proceeding toward the efficacy part, we need to carry out the dermal toxicity study for the safety aspect. Here we have chosen the OECD guideline because this guideline, promotes the minimal use of the animals. So therefore, we have used this guideline, and the animal we have recruited is sprag dolly rats, healthy adult sprag dolly rats. Then in the last portion of my slide, we will understand, we will discuss about the wound healing efficacy of this ethyl pyruvate based hydrogel from the point of view of its use for prophylactic as well as for mitigation. And why prophylactic? In case if in any radiation emergency occurs and I need to we need to send some rescue operational personnel or emergency responders, then we need to provide a solution in form of prophylactic use. But in case of persons who have been already exposed and they are termed victim, then we need to apply the cream after the exposure. So it is called mitigation. So from the both the point of view of emergency responder and victims, we have tested our cream for its efficacy. So now let's move to the first slide of the presentation, and I am going to start with the how ionizing radiation cause protein damage at the molecular level in solution. So what we have selected a bovine serum albumin compound, which is a very well characterized protein, and we have radiated this protein in saline with the ionizing radiation, which is we have used the gamma source GC 5000, and with the dose of 500 gray and 1000 gray. Now, how this figures this show that how the protein is getting damaged. So we can see that the BSA has a molecular weight of around 65 kilodaltons, and in the natives, we can see that there is a bright band for corresponding to the 65 kilodalton protein marker. So this band shows the presence of protein. Now, as I radiate this protein to 1000 gray, so we can see that there is a feeble, a very faint band corresponding to 65 kilodalton. So the decrease in the intensity of the protein band in the SJ space shows the damage in the protein, native protein. And similarly, we have seen in the 500 gray, the intensity is decreased, but it is not as much as the happen in the case of 1000 gray. So both the radiation dose, the edge space profile of PSA is showing that protein is getting damaged. But in presence of drug, ethyl pyruvate at three different concentration from 15 micromolar, 150 micromolar, and 1500 micromolar, the brightness of the band, the intensity of the band is more than that of the protein sample which has been exposed to 1000 gray and the 500 gray. So this shows that the ethyl pyruvate is able to protect the BSA against the oxidative damage of the protein. We have also carried out the control studies for the ethyl pyruvate uh, of the BSA molecule, which has been treated with the ethyl pyruvate without the presence of aniline radiation. So both the positive negative control we have taken care in this as this profile. Now we have moved to the spectroscopy to support this data of SJ space. So we know that from the literature that as the protein get radiated, then there is an enhancement in the absorption capacity because of the changes in the conformation of the protein. So we find the same case in the from this is the dark band is from BSA control, and for the one, and this is the band for the 1000 1000 gray exposed protein. But when we treat these molecules with the protein with this ethyl pyruvate prior to the irradiation, and then we expose it, then the enhancement of the radiation of the sorry absorbance is in between the native one and the full irradiated protein sample. So this show again that the conformation change is there, but it is not as much as in the case of exposed protein. Then we conducted the fluorescence measurement of the irradiated protein in the with and without the presence of ethyl pyruvate. So there are two important marker there. One is biotyrosine formation, because in the theory part it is well mentioned that due to the presence of multi-center radicals in the polypeptide chain, there is a chance that the protein may get aggregate, and because of the aggregation of the protein, or there is a chance of formation of biotyrosine. It radiate it emit around fluorescence intensity around 420 nanometers. So this is a marker for the protein damage. Similarly, for the tryptophan, which is one of the aromatic protein of the bovine albumin. So if there is a damage in the protein, so there would be a change in conformation, and because of change in conformation, the fluorescent intensity emission due to the tryptophan got compromised. So there would be pinching in the fluorescent intensity of the tryptophan emission intensity. So in case of the radiated one, for the biotyrosine formation, it would be high. Like for example, in this you can see that for the control, it is a black one. So it is around 4,000 counts. But if you radiate it with 500 gray, then you can see that it goes to near about 9,000 counts. So there is a formation of, or there is a indication of protein damage. But those samples which have been treated with ethyl pyruvate prior to radiation, and then we expose them to the 500 gray, both in the case of 1000 and 500, we can see that these bands fall in between the native protein and the fully or the radiated protein without treatment of ethyl pyruvate. So this indicates that yes, the damage is there, but the damage is not as much as only in the irradiated DSA. Similarly, for the tryptophan, if the for the control, it is around the count is around 20,000. But for the sample which has been irradiated with the 500 gray, the counts comes near to 5,000. So there is almost 1/4 change in the emission intensity. So now we can see that for those being a protein sample we treated with the ethyl pyruvate with different doses of 15, 150 or 1500 micromolar, the damage is there, but the damage falls in between the native protein and the sample which is irradiated without ethyl pyruvate. So in both the cases, it being well understood, the damage part is there, but the damage is not as much as only in the case of the irradiated bovine cell albumin. So the data suggested that ethyl pyruvate is protecting bovine cell albumin against the protein damage. Then if the some confirmation is taking place, then it is very important to undertake the circular dichroism spectroscopy and the melting studies of the VSA. So what this basically the data suggesting in the CD spectroscopy, there are two prominent band. One is at 222 nanometer, another is at around 209 nanometers, correspond to the alpha content of the protein. So we can see that in this spectrogram spectra, there is a band around 222 and there is a band on 209 for the native proteins. So I think everybody can see it. So this basically characterizes that yes, it is a the protein with the alpha content. Now, if you radiate it with the around 500 gray or 1000 gray, then the band, bands goes up, and it lead to the decrease in the intensity of the band. We can see that. But those samples which are being treated with ethyl perlite prior to radiation falls in between the two bands. So therefore, again, it supports the data as we have seen in the case of the UV spectroscopy and the fluorescence spectroscopy. Similarly, we find that if the protein get conformation change, whether it will result into the enhancement or decrease in the protein melting. Because protein melting is basically indication of the mechanical strength of the protein. Now there is a change in the melting temperature, but this again, those being treated with the ethyl pyruvate prior to the radiation, there are the changes, but changes are not as much as in the case of the treated ones. Then we have done some biochemical assays. Like I told you before that there are two important parameters. One is the carbon formation, because in the damaged proteins there are good content of carbonylation. So we can see in this figure that uh, in the native protein, the chance of getting carbonation is 10 micromolar. But if you irradiate it, then the carbonation become 70 to 90 micromolar depending upon the radiation dose. But for those who's been treated with the ethyl pyruvate, the content is less as compared to the irradiated one. For example, if I compare with the D3 dose for the 500 grays, the for 500 is 70 micromolar, and for the D3 it is around 50 to 60 micromolar. So it indicating the protection aspect of the ethyl pyruvate. Similarly, we have done for thiol, because we know that the thiol content is it is because the thiol content it indicate the stability. It indicate basically the the content of the protein. So if in the native it is 60 micromolar, in the case of radiation it becomes 40 micromolar. So it means that you are compromising the thiol content of the protein. So here again, it again showing that ethyl pyruvate is protecting the protein. So we got this paper published in International Journal of Biological Macromolecules in 2020, and now its impact factor is around 6.7. So it's a very um, well cited paper now, and many people are taking reference for their further studies. Now let's move to the DNA damage. And again, DNA damage, because I told you before that it's important organelle, and it is the storehouse of the hereditary information. And if you say that everything is governed by the DNA. DNA is very important aspect of for the human survival, and therefore it is very important to understand that how the radiation causing damage to DNA at molecular level. So for this, we have chosen a plasmid DNA for the for the electrophoresis study, and for the spectroscopy study, we have chosen the the calf thymus DNA. So the calf thymus DNA is a well characterized and well recognized and well accepted DNA. If you can do some study with calf thymus DNA, then nobody question your studies. Then we have started with the circular diagram, because DNA is also a stereochemical compound, and we expect there will be a optical rotation. So there would be a change in the circular diagram signal. And we have a two prominent band in DNA, one for 275 nanometer and one at 245 for different form of DNA. Then what we have seen that in when you radiate the DNA prior to the recording the series spectrograph, then there is a changes in the band intensity. But if you know treat the DNA with the ethyl pyruvate prior to irradiation, then the intensity falls in between the native DNA and the one which is aerated without ethyl pyruvate. And we have also done the CD study. We can see that here, the melting study. In case of calf thymus DNA, it is 81. So when you increase the in the case of ethyl pyruvate, then again there's a decrease in the 2 degree in the melting uh, profile of DNA. So therefore, it's suggesting that yes, there's some changes in the conformation of DNA on interacting with ethyl pyruvates. And we have also shown through the UV spectroscopy and the melting profile of UV that yes, in the ethyl pyruvate is interacting with DNA, because it is important. It is written in many research papers that if a small compound is able to interact and binds with the DNA, then it enhances the structural integrity of the DNA. So therefore, we have done this. Then we have performed the antioxidant assays on the ethyl pyruvate. So one we have done with the DPPH, FRAP assay, and the FRAP assays and DNA both shows that at higher concentration, like for example 5,000 micromolar, the it is a very good radical scavenging properties, and it able to through the MDSA, it able to scavenge the radicals which has been generated in the reaction condition. Then we have done the docking studies to understand that how this molecule dock, and what is the possibility of is whether it is going to dock in the minor group region or the intercalate as a intercalator. So we have done both the things, and we find that the minor group binding is more preferable as compared to the intercalation. And so this molecule is perfectly binds, and one of our previous studies showed that this compound is binds with bovine serum albumin as well. So this is a good prominent molecule, and that is why because of anti-radical scavenging and expanding with the macromolecules, biological macromolecules, it's enhanced. It able to save the bovine serum albumin as well as the calf thymus DNA from the radiation-induced oxidative damage. Now let us move to the next slide. And so we have what we have done with the plasmid DNA. We have run the plasmid relaxation assay, and we have exposed the DNA to different doses of proteins. Then we have selected that yes, at 50 gray of dose is the optimal dose where we can see some damage. So I will explain you like this. In the first lane, we have a native DNA, which is mean that it is the one which has been not exposed to radiation. And we can see two bands, one for the supercoiled, and again is a very very light band for the open circular form. So there is no L form band here, no linear DNA. But if you increase the, or you can say that if you expose to 50 gray of ionizing radiation, then the supercoiled band intensity decreased, and the band which is open circular form, got intensified. So, and and the slight, feeble intensity band corresponding to linear form of DNA, appears in between them. So this is the indication of the damage in the DNA structure. And if you increase the radiation dose, then your overall your supercoil form is completely goes off. So here in this structure, we can see that the direct manifestation of radiation induced damage in the plasmid DNA. Now we can see that if you give 200 micromolar or 500, 750 or 1000 micromolars of different doses of ethyl pyruvate to DNA prior to radiation, and then you expose them to the radiation, then you can see that the there is a decrease in the intensity of radiation, but the decrease is not as much. So you can see here, the intensity is less as compared to native one, but it is more intense than the one which being exposed to 50 K. And similarly, like if you move to 5,000 micromolar of ethyl pyruvate, you will find only the supercoiled band, no L band, and very feeble OC form. So again, if you compare with one and the 10th lane of this, plasmid relaxation assays, uh, profile, then you can see that it is almost same. So therefore the 5,000 micromolar is able to withstand the radiation induced oxidative damage. We have done some derivation of this data in form of bar diagrams. So you can see from this bar diagram also the native and the 5,000 gray is almost same. Similarly, we have done for the the different percentage wise we have designed the data. Like for example, you can see here, like if you increase the radiation dose, like for example to 50 gray, then the L form is around 10%, and with the supercoil form it is around 80%. Fine. And around 80%, and the open circle form is around 10%. So this is the percentage wise we have done. So now let us move to the objective number two. So before going to the objective number two, we will shall have some break so that you can rejuvenate yourself, and then we will take this formulation two and the other part. Thank you.